Hello everyone, and thank you for joining me for our webinar titled Advances in Extracting and Stabilizing RNA, an important toolkit for molecular oncology and immunology. My name is Jeff Chapman, and I am the CEO of MicroGEM International, a biotechnology company whose primary focus is towards the democratization of molecular biology. Like most of you, I am presenting this today from the comfort of my home office, as we manage our way through this challenging global pandemic. I'll make a promise to you today to spare you from the reminders of SARS-CoV-2. However, with one exception, and I'll state it now, that the extraction techniques we discuss today will work equally well for this virus. Further, in my use of the word we today, I am referring to the royal we as MicroGEM, as I am merely given the honor of presenting this to you today. We'll begin this talk with a short introduction on MicroGEM, a company which may be new to many of you in the audience. I will touch on the challenges involved with isolating RNA, most notably the stringent measures required to minimize contaminating RNAses. I will then shift to one of our core technologies, which is the enzyme-driven RNA extraction modulated by temperature. I will share with you some examples of how our customers have implemented this approach in molecular oncology and immunology, and I will speak to its scalability from a single cell to hundreds of thousands of cells. To wrap up this presentation, I will provide you with a little peek into our future, outlining a fully integrated gene expression system that we are developing for point of need. Finally, I will conclude with a summary and acknowledgements of all those involved in the fine work I'm presenting to you today. Well, our name's MicroGEM, and this name and logo truly speak to our core competencies as a biotechnology company. In our case, micro is in reference to our expertise in microfluidics, both internally developed and through collaboration with the University of Virginia and the University of Otago. GEM, on the other hand, is an acronym standing for genes, enzymes, and microbes. And this references our focus on gene expression for assay development, our expertise in enzyme discovery, and of course, our accumulation and curation over a diverse collection of microbes discovered across New Zealand and Antarctica. The company began under the name of PacificGEM, a New Zealand biotechnology company founded by a group of very adventurous Kiwi molecular biologists, enzymologists, and mycologists who went off on explorations across Antarctica and New Zealand on a quest for microbial diversity. These photos are just a few examples of some of the far reaches they went to on their search. In the photo on your upper left, you can see the hut of Sir Ernest Shackleton, a very famous Antarctic explorer who led 3 British expeditions to the Antarctic. From these historic sites, we were able to gather many interesting cultures, from things such as old horse leather harnesses, to left behind food scraps, to mutton and seal carcasses, to the roof of the nearby latrine. In the distance beyond the hut, you will see the majestic Mount Erebus, the most active volcano in Antarctica. Our exploring scientists scaled this mountain as part of their search. In the middle upper photo, you can see a geothermal vent at Lake Taupo. This is on the North Island of New Zealand. The orange color that you see is really signifying the presence of bacteria from the genus Thermus, a great source of thermophilic extremophiles, no doubt. And in the upper right photo, you can see the McMurdo Dry Valley. These valleys have remained ice-free deserts for more than 5 million years because any precipitation that falls as snow on the valley floor is immediately blown away by strong, dry, katabatic winds through a process that's known as sublimation. The result, of course, is the world's most extreme desert. Astonishingly, though, just inches below the surface, it's teeming with microbes, clearly having evolved to grow in what is essentially an anhydrous environment. Finally, in the middle lower photo, you can see our Chief Scientific Officer, Dr. David Saul, taking up residence on the Ross ice shelf, once again in search of microbial diversity. These explorations resulted in the accumulation of an extensive and diverse culture collection ranging from psychrophilic fungi to hyperthermophilic archaea. This collection has become a treasure trove at MicroGEM for the discovery of novel enzymes and metabolites. Although we began as Pacific Enzyme Company and later evolved into Zygem, we passaged through a series of acquisitions and consolidations to become what is now MicroGEM, a global biotechnology company headquartered in Southampton, UK, with R&D sites in Charlottesville, Virginia, and Dunedin, New Zealand, and manufacturing in Guilin. Our vision is truly as what is stated here. It's to democratize molecular biology through the development of point-of-need solutions to solve complex biological problems around the world. We will achieve this through the seamless integration of sample preparation, molecular assay development, and molecular analytics like RT-qPCR. Although this sounds straightforward, believe me, it is rife with challenge and, of course, with opportunity. In today's presentation, we will focus on sample preparation, and we will finish with an example of our progress in integrating all three critical processes. For sample prep, of course, it's all about simplifying the process. For molecular assay development, it's about reducing complexity to enhance robustness. And for analytics, it's really about reducing footprint to being a person-portable device while maintaining performance. And of course, wrapped around all this is a predicate design with no or virtually no moving parts. However, in order to democratize the analysis of RNA, we began with sample prep, as this is one of the most difficult challenges. What makes this so challenging is the traditional extraction methods of RNA require a laboratory, and not just any laboratory, one that is designed and heavily equipped for RNA extraction. In order to integrate sample prep, we will need to innovate and really rethink how nucleic acids are extracted. Of course, to further add to the challenge, RNA is highly sensitive to degradation by RNAses. And yes, RNAses are indeed frightening. This is due to their chemical nature or their chemical structure. RNA is much more susceptible to degradation than is DNA. And RNases can be tough. They can be resistant to metal chelating agents. And some, like RNase A family, can even survive prolonged boiling or autoclaving. It is fair to say that this has created a healthy paranoia in those taking up the challenge of doing RNA work, because contaminating ribonucleases can be found everywhere, originating from flakes of dead skin laying on the lab bench, fallen human hair getting into your experiment, even pet hair that maybe you brought in the lab from your clothing. Transfer of perspiration and skin oils can occur simply by touching your face, even with a gloved hand. And of course, the most abundant source of ribonucleases, contaminating ribonucleases, is the presence of bacteria and fungi, either being tracked in, blowing through an open window, or circulating through the ventilation. There's no getting around it. Contaminated ribonucleases can be found everywhere. The appropriate response, of course, to this has been to introduce countermeasures of precaution. These include things like double gloving your hands, treating your water and buffers with diethyl pyrocarbonate, dedicating tools like pipettes and glassware for RNA use only, designating RNase-free zones to counter contamination, baking your glassware at 200 C for at least 4 hours, decontaminating polystyrene and polypropylene devices like electrophoresis tanks with hydrogen peroxide, and of course, purchasing tips and tubes from vendors who have certified that these are nucleic acid free. All of these points really point to a need to rethink how RNA is being extracted. As with many methods, They begin with what works, and that gets passed along. And then a process gets improved over time. With the isolation of nucleic acids, the critical link has always been at the first step of cell lysis and protein denaturation. Here we have historically incorporated detergents or chaotropic salts. And this one step has driven multiple steps thereafter in order to remove those agents offensive to further molecular analysis. Great place to start. If we're going to be effective with this journey of integration, our sample preparation needs are going to include the ability to disrupt tissues and lyse cells without the use of detergents or chaotropic salts. We need to hydrolyze proteins within nucleoprotein complexes. We need to destroy all endonucleases and exonucleases from both the sample and contaminating environment to prevent degradation and to stabilize the RNA. We need to be able to retain all types of RNA molecules from very small to very large species. This, of course, can be tricky when purification systems are employed. We need to be able to eliminate external contamination, and we need to have compatibility with the integrating RNA assays, or what's the point, right? Finally, we need to complete the entire process in less than 15 minutes. Although these are very daunting set of user requirements, our achievement of this subsystem will immediately bring value to your lab that you can use in tandem with existing molecular techniques, while forming a key subsystem for our future development of integrated devices. Our breakthrough towards this objective came from those explorations to those early explorations to Antarctica that we spoke of earlier in this presentation. Of course, or specifically, they came from a discovery of a thermophilic bacteria, which was collected from the volcanic vents on Mount Erebus, where we isolated a novel enzyme which became known as EA1. This enzyme had all the characteristics we were searching for. It was a neutral proteinase, making it compatible with buffer systems that we typically use for RT or PCR experiments. It was highly thermostable, giving it longevity in an assay system or for storage. Most important though, it has a temperature optimum at 75 degrees Celsius, where proteins are already denatured, allowing it to simply rip through notoriously recalcitrant proteins like RNAses and nucleoproteins without the need for denaturants like SDS. At the same time, this enzyme has practically no activity below 50 degrees Celsius, allowing it to cohabitate happily with other enzymes until it is activated by temperature. This enables temperature to be used as a switch to turn enzymes on or off, which is truly at the heart of our RNA-GEM extraction methodology. By simply adding RNA-GEM to your sample and raising the temperature to 75 degrees Celsius, we are able to lyse the cells, destroy all nucleases, and remove the nucleoproteins which would interfere with downstream assays. This process can literally be carried out using a heat block or a thermocycler and can be completed in 5 minutes. Depending on the RT and the polymerases that you plan to use in your assay, you may choose to add an additional 5-minute step at 95 in order to denature the RNA-GEM to prevent the hydrolysis of other enzymes that may come after. What you are left with ultimately after this reaction are total nucleic acids, clear of nucleases and ready for RT and PCR. As everything else is done within a single tube, all species of RNA are retained, including short, non-coding RNAs, in what essentially is a single-step extraction. This met our requirements criteria, and we believe is ideal for integration, whether within your RNA analysis workflow today or within our future microfluidic devices. Of course, if you're using intron-spanning primers, the presence of total nucleic acids can be valuable, as you can normalize your gene expression to the DNA, kind of acting as a surrogate for the number of cells in your sample. In cases where you may not be using intron-spanning primers or are concerned with splice variants or working with microbes which do not contain introns, then the removal of DNA will be required. This can be accomplished through the simple addition of DNAs followed by a short incubation step. And of course, waste not, want not. Relative to other RNA extraction methods, very few tips and tubes and reagents are consumed. As you scale to hundreds and perhaps thousands of samples, this generates a sizable cost and carbon footprint reduction. So what about those RNAses? Did we really destroy them as we say? To test the efficacy of RNA-GEM at removing contaminating ribonucleases, we challenged it with the worst case scenario. We incubated our RNA with large quantities of RNase A, one of the hardest ribonucleases to destroy. We dual labeled a synthetic oligo with a fluorophore and a quencher, such that if the RNA was digested by an RNase, the fluorescence would increase. In the reaction, we deployed RNA-GEM at the concentration we use for extraction. We placed that in the presence of the dual labeled RNA, followed by the addition of increasing quantities of RNase A. From the graph in the slide, you can see that essentially no RNase activity was detected up to and including 0.1 Kunitz units, a quantity well above the functional level that you would deploy in an RNase protection assay or a plasmid purification where you're trying to rid the sample of all RNA. This gave us great confidence that the presence of RNA-GEM would wipe out any background contaminating ribonucleases in your lab, which are present at levels well below 0.1 Kunitz units of activity. Although we're not suggesting to relax good laboratory practices, I hope this data demonstrates to you that the paranoia of contaminating ribonucleases can be relaxed. Certainly it seems to be the case for this stock photo actor who is happily wearing goggles to view the computer. However, this approach really should go a long way to improving the robustness of gene expression assays and towards improving the accuracy and precision of gene expression quantitation. However, as my grandmother used to say, the proof is always in the pudding. In this experiment I'm showing you here, HeLa cells were collected and counted and serially diluted to create 9 samples with cell numbers ranging from 10 to 50,000 cells. These cells were then extracted using both the RNA-GEM method and a trizol method. 5 microliter aliquots from these extracts were then deployed in an RT-qPCR assay to quantify the expression of both a high abundance gene, beta-actin, and a lower abundance gene, BRCA1. Gene expression of both the higher and lower expressing genes generated a perfectly linear response from 10 to 50,000 cells when using the RNA-GEM extraction protocol, while the alternate methods seem to be getting a much poorer recovery of RNA, especially at the lower abundance levels, as seen by the lower response and non-linear response. In fact, the single tube enzymatic approach has already been shown to work well, even for a single cell. In 2015, a study published in the Journal of Circulating Biomarkers by Hoshino et al. Featured a single cell RT-qPCR method that they had developed for rare circulating tumor cells. This was literally a 5-minute extraction using only 0.2 microliters of the RNA-GEM enzyme. They analyzed 10 genes from a single cell isolated from blood where they had spiked in cells from 3 different breast cancer cell lines. The result from a single cell correlated well with those from 1,000 cells, with the exception of markers like HER2, which showed differences from that of the larger population. For MicroGEM, the use of RNA-GEM and PrepGEM for single cell extractions has really become one of our more significant parts of our business. As the use of microRNAs and exosomes have shown great promise as potential therapeutics and delivery vehicles, we often get inquiries about whether this enzymatic approach would be compatible. We highlight here a publication from as early as 2015 from the group of Almanza and Zanetti, who demonstrate the effective recovery of microRNAs from exosomes created from transfected J558L cells. As the reaction is carried out in a single tube, there are no purification steps, so there are really no points of loss for these smaller RNAs. To evaluate the simplicity and ruggedness of this RNA extraction methodology, we asked one of our MicroGEM scientists whose focus has been on microfluidic chip design and who had not had familiarity with this kit to incorporate this method of RNA extraction in an experiment he designed with his wife at the University of Virginia. With no instruction, he simply took the kit and followed the protocol. The model they chose was the macrophage-like raw 264.7 cells using LPS to induce the expression of pro-inflammatory genes. As the primers they were using were not intron spanning, they chose to add a DNA step. However, as these particular cells appeared to burst upon contact with the lysis reagents, they reversed the DNA step by putting everything together in the same reaction mix at the start. As RNA-GEM-mediated lysis was not required in this case, they began by ramping to 37 degrees to activate DNAs to remove the DNA. Then by simply ramping to 75 degrees, they activated the RNA-GEM, which destroys the DNAs and all of the nucleases and any nucleoproteins, essentially leaving RNA behind in a soup of nucleotides, peptides, and amino acids. The results were as we hoped. With the stimulation by LPS, both IL-6 and TNF-alpha genes significantly increased in expression and compared well with the traditional methodology that the laboratory was previously using. This method provided, or this experiment really, provided verification for us that the protocol was easily transportable and demonstrated nicely the use of temperature as a switch to create stepwise enzymatic functions as part of the sample preparation process. We also found the scaling of this approach to RNA extraction surprisingly straightforward. When working with either a single cell or a small number of cells, one simply needs to reduce the amount of RNA-GEM used in the reactor. We've been selling this subsystem of our sample preparation process as RNA and DNA extraction kits for several years now, kits which you can readily adapt into your RNA workflow. Now with the chemistry component of this in place for our sample prep, we shift our focus to the next step, which is to make this all portable. In other words, taking the next steps towards the process of moving RNA extraction outside of the laboratory. The solution was found within the design of a sample preparation cartridge, in which the sample could be placed with our chemistry in a sample chamber made from a thermal-responsive polymer that will shrink when temperatures are raised above 80 degrees Celsius, along with a heat burstable valve, which will open at the same temperature. In doing so, we can incubate the sample at 75 degrees C and let the enzymes do their magic, then simply ramp to a higher temperature to open the valve and allow the heat shrinkable tube to drive the contents through a short purification column, which captures the RNA-GEM enzyme as well as unwanted inhibitors, allowing the nucleic acids to flow out the bottom. An example of this was demonstrated by the group of Joe Stanton from the University of Otago, whom in collaboration with Linda Boykin from the University of Western Australia, took this nucleic acid extraction process to several cassava farms across Africa to test for the cassava virus. This work was done in combination with the MinION sequencing system to identify the cassava virus literally under the shade of a tree on the farm. This is truly point of need. To evaluate this approach further, we developed a portable device which could contain up to 24 cartridges at a time for extraction into strips of PCR tubes. We call this device the PDQ-EX, Nucleic Acid Extraction System, which internally and externally now stands for pretty damn quick extraction. This system essentially works through cycling the temperature for the enzyme system to extract and elute the samples automatically. Although primarily used commercially within the laboratory, it can be battery powered and has been used remotely to take extraction to the source of the sample. Our next step in this journey has been to integrate the RT step and the qPCR step with the sample prep. To accomplish this, we have been developing a microfluidic chip in which the RNA flowing from the sample can be incubated with reverse transcriptase to create cDNA, which will then flow into one of 8 different wells where the primers and polymerases are waiting for amplification and detection of the genes that you may wish to quantify. The resulting system will be a person-portable, battery-powered RT-qPCR system, which will make and take molecular testing to the point of need. That's our focus. From our preliminary work, we anticipate eliminating a lot of the gene expression variation that is due to the prolonged nature of first capturing a sample to then processing it. We would be very interested in hearing from you who have gene expression or may have a gene expression panel that would be ideally suited to move the point of need. In summary, what I've shared with you today introduces the concept of temperature and enzyme-driven extraction of RNA. And I hope that I've demonstrated to you that this approach is very fast and may be accomplished within a single tube with a single step. This, of course, maximizes recovery and minimizes contamination. I hope I've convinced you that the method is easily scalable from a single cell to hundreds of thousands of cells, and that RNA-GEM effectively degrades RNAses, stabilizing the RNA and simplifying the management of RNA extraction. This method extracts all RNA, including non-coding RNAs like microRNAs, and that this approach is more environmentally friendly, significantly reducing the amount of plastics and reagents used. Of course, important for us is that it be fully integrated with microfluidics, and I believe we hopefully have demonstrated that to you, paving the way for point-of-need gene expression systems that anyone can use. This all goes towards our vision of democratizing molecular biology. As I mentioned at the start of my presentation, although I've had the honor to present this work to you, my many colleagues and collaborators were the ones who have actually accomplished this work. I would like to acknowledge some of these key members of our MicroGEM team who worked on this, including but not limited to David Saul, Abhishek, Grant Broomfield, Jeff Hickey, Orion Scott, Jingyi Li, Patrick Collins, Giuseppina Sanino, and Leanne Landers. And of course, our emeritus explorers from PacificGEM, who include Roy Daniel, Hugh Morgan, and Roberta Farrell. Our key collaborators from the University of Otago, that includes Joe Stanton, Chris Rowell, and Jackson Treese, and our key collaborator at the University of Virginia in Charlottesville, James Landers. Thank you so very much for your attention. And now I would be pleased to entertain your questions. Thank you very much.